The cell cycle is a series of events that happen during the lifetime of the cell until it gives birth to two new identical cells. First is the interphase, which is divided into G1, S, and G2. In G1, which is the longest part of interphase, happens the growth of the cell. In S happens the DNA replication, and in G2, the cell prepares itself for cell division by making a second copy of all its organelles. This is the first stage of mitosis called prophase. In this stage, the chromosomes consist of two copies each, called sister chromatids, and become visible to the microscope and are attached by a centromere. Also, the centrioles move to the opposite sides of the cell, forming microtubes which form the spindle fibers. Later on, the nuclear membrane breaks, freeing the chromosomes. This is the second stage of mitosis, metaphase. In this stage, the spindle fibers cover all over the cell, and the duplicated chromosomes attach their centromeres to the spindle fibers, and also align themselves in the middle of the cell. This is the third stage of mitosis, called anaphase. In anaphase, the centromeres split and each sister chromatid is pulled apart and moved to the opposite pole of the cell. In late anaphase, the chromosomes have reached their respective poles and the microtubules begin to disappear. This is the fourth stage of mitosis, called telophase. In telophase, a nucleic membrane is formed around each set of chromosomes, and the cell begins to constrict so it is ready to separate. This is the only missing step, cytokinesis, where the DNA becomes invisible and it gives birth to two new identical cells by the separation of the cytoplasm.